Okay, guys, the next big topic in this unit is protein synthesis. And we're going to go over really briefly how protein synthesis works, and we'll probably talk about it a lot more in another unit. But let's just go through and get the big ideas from this. Um, well, we want to talk about how our proteins are made. And remember, we talked about enzymes before, and we said enzymes are a really important protein because they carry out a lot, they help us do a lot of these really important metabolic reactions. But proteins are super important structural molecule, as well as being enzymes. They give our cells structure. They help move stuff around. They help hold stuff in place within a cell. They're really important with muscular development. The list of the uses and functions of proteins goes on and on and on. So they're really, really important. Everyone's set of proteins, when they're turned on, when they're turned off, how they influence your metabolism, that is kind of what makes you different than everybody else. Your protein synthesis is directed by your DNA, which is unique to you. Now, the DNA, deoxyribonucleic acid, there's two types of nucleic acids we're going to look at, is made up of a monomer called a nucleotide. And I'll show you a picture of that in a second. The DNA is made of nucleotides, and then these DNA sequences are organized into genes, and those genes are organized into chromosomes. So there's a kind of a layer to it. And if you look at the picture on the side there, you'll see at the top part, we're actually looking at the nucleotides of the DNA. So these C, T, G, and A, those are nucleotides, and they join together to form what we often call the rungs of the ladder. So DNA is often compared to a ladder. You get the backbones that go on either side, and then you get the rungs that go in the middle that connect the backbones together. Well, these CTs and Gs and As, these are the ones that connect the two ladders together. Then of course the ladder twists, and then we get that structure that everyone hopefully is familiar with in DNA. But DNA is a really long strand of these repeating or sequences of these nucleotides. So what happens is the DNA actually starts to twist up, and when it twists up, it gets shorter and thicker, and it goes from long stranded DNA like this that we often call chromatin into something that we call a chromosome. And chromosomes we're going to talk about in the last part of this unit, which is cell division. Now, RNA, ribonucleic acid, the other form of nucleic acid, that has a major role to play in protein synthesis, but it doesn't form part of chromosomes, and it doesn't have a clear makeup or a part of your genetic makeup. And we'll talk about this stuff in the next few slides. Now, in order to understand protein synthesis, you have to have an idea of how the nucleus in the cell works. So the nucleus is often called the control center of the cell, and the reason why it's called that is because it has the DNA, it has the chromosomes. And on those chromosomes are the genes that direct every cell function in your body. So the DNA is the blueprint that makes you, and it makes sense of how you work, how you function. So inside the nucleus, we're going to have all of this, and you can see here all of these pinkish lines. That's all going to be chromatin, and all chromatin is long strands of DNA. And 99% of the time, DNA is in the form of chromatin. It only presents itself as chromosomes during cell replication. So only for a very short period in the cell's life does it show up as chromosomes. So we've got our nucleus. The nucleus has a membrane around it that's often called a nuclear envelope. And inside the nuclear envelope, you're going to have the chromatin, which is the DNA. And you're also going to have an organelle here called the nucleolus. Now, the nucleolus is an organelle that creates ribosomes. And ribosomes are the site of protein synthesis, and we'll talk a little bit more about that right now. All right, first let's have a look at DNA. So let's start over here on the right hand side first. So this is one single nucleotide. And for DNA, this is going to be, the nucleotides are going to be the monomer. So each monomer, each nucleotide is going to be made up of a nitrogen base, and that's going to be either adenine, thymine, cytosine, or guanine, or cytosine and guanine. So in DNA, you'll often see A, T, G, and C. Adenine and thymine pair together, and guanine and cytosine pair together. So these patterns of ATs and GCs, this is what makes you and gives you your unique structure. So here's our nucleotide. So here's our same nucleotide right here inside this square, and you can see it pairs up with another nucleotide on the other side, and this repeats over and over and over and over and over. The sugar unit and the phosphate unit is going to make up the this part of the molecule, and the nucleotide is going to make up the parts that go in between it. Okay, so the DNA twists and twists and twists and twists until it does, and it forms into what we call a chromosome. So a chromosome is a short, thick chunk of DNA where the chromatin has twisted and twisted and twisted, almost like a charging cord or an old telephone cord. Once it twists up, it turns into knots. Now that's exactly what happens to chromatin. It twists itself up and it turns into a chromosome. Now the different parts on each chromosomes, and they got them here in different colors, those are going to be a gene. And generally what a gene is it's a code to produce a protein. So it's an instruction manual on how to produce a protein. So each one of our genes will produce proteins that will contribute to our eye color, or how tall we're going to be, or a whole bunch of different metabolic functions, because remember, it controls the enzymes, it controls your cell structure, it controls musculature. I mean, so much stuff is dependent on proteins. It makes them a majorly important molecule. Okay, so let's compare DNA and RNA. So these are the two types of nucleic acid that are involved in protein synthesis. DNA, first of all, is found just about completely inside the nucleus. You don't find DNA outside of the nucleus. Now RNA, on the other hand, is only in the nucleus for very brief periods of time. So you almost always find it in the cytoplasm. The composition of the monomers for DNA, the sugar molecule that we talked about, is a sugar called deoxyribose, and the nucleotides are going to be adenine, guanine, cytosine, and thymine, like we talked about before. So the AT and the GCs. If we look at RNA, that sugar is not going to be deoxyribose, but it's going to be ribose. Okay, so deoxyribose is where we get the D, and then ribose is where we get the R. The nucleotides are going to be slightly different. In RNA, we still have the guanine and the cytosine. They're going to pair together. We also still have the adenine, but we don't have thymine. Instead of thymine in the RNA, we have uracil. So those are slightly different nucleotides that make up RNA. The structure of DNA is what we call a double-stranded helix, and that's that twisted ladder look that most people would recognize if they thought of DNA. So when we say it's a double strand, it's because it's two sides. The ladder's got two sides and the rungs that go in between the ladder. Now RNA is a single strand. So instead of having our ladder structure like we have for DNA like this, RNA is going to be like this. We still get the sugar phosphate backbone here. We get the nucleotides right here, but we don't have the other side. So that's the difference in the structure between DNA and RNA. The function now, for DNA, this is going to be the hereditary control center of all cell activities. Okay, DNA makes up the chromosomes. The genes carry the sequence of nucleotides, or the code in the nucleotides, that give every cell in the body its instructions on how to make proteins. That's the function of DNA. Now, RNA is there to take that code from the DNA and turn it into proteins by using 3 different types of RNA. First, we're going to have messenger RNA, and then we'll have transfer RNA, and then there's another type of RNA called ribosomal RNA. So let's have a look at those different versions of RNA. Usually what we do is, when we're talking about RNA, we just put the small letter in front of it. So messenger RNA becomes mRNA, ribosomal RNA becomes rRNA, and then transfer RNA becomes tRNA. So let's start first with messenger RNA. Now, messenger RNA is actually put together on a strand of DNA. So inside the nucleus, this is where the mRNA is formed, and it makes itself a copy based on a section of DNA that has that gene, that code for a protein. So what the mRNA does is move into the nucleus. It forms based on the template of the DNA, and then it takes that formed piece of mRNA and leaves the nucleus, goes into the cytoplasm, and attaches to a ribosome. That's what mRNA does. The big function, the process that it does, all that is called transcription, and we'll give a little bit more detail to that in a couple of seconds. For rRNA, now ribosomal RNA, these are the proteins that actually make up the ribosome. Now, ribosome is a really important organelle. It's not membrane bound. It doesn't have a membrane surrounding it. And it's the site of protein synthesis. So proteins are made at the site of, they're made actually on the ribosome. So that's really, really important. But our ribosomal RNA, it makes these proteins that help make up the ribosome. And the third one is the tRNA. Now, the tRNA works with other forms of RNA, mostly mRNA, and it takes the code that was produced by the mRNA that was transcribed from the DNA, and it brings along amino acids. The tRNA also helps form a peptide bond that joins amino acids together. Now remember, proteins are long chains of amino acids. So now we'll get into how these 2 different processes work.